Okay. Lecture 16.
Okay, so today begins sensory physiology. The whole book was non-subject. Really interesting subject. So we'll spend the next three lectures on sensory. The first one today is called general sensory, and then I'll finish it on Tuesday, and then we'll have a lecture and a half on what's called spectral sensations. So it's a very interesting topic. After class in the room behind us on 24, if you haven't had a chance to review your unit 2 exam, you're interested, we'll do that. And the quicker questions is the beginning of today's lecture. So go ahead and do that first. So if you want to get your transponders out, turned on, first question. Which of the following types of glial cells serves as a macrophage in the central nervous system? Just one a simple little memorization question. Okay, 4 seconds. Numbers have gone down quite a bit since the drop period, looks like. Okay, and wow, 90% of you got it. Almost everybody in microglia is right. You should know the major things each of these cells do and a little bit more detail about astrocytes play a pretty significant role. Question 2. Early histologists named dark staining masses within the soma nissl substance. Today these structures are known molecularly as okay, 10 seconds, 5 seconds, okay, and 92% again, so sounds like you're keeping up pretty well. Question 3. Neurotransmitters synthesized in the neuron cell body or perikaryon are transported to the axonal ending along which class of cytoskeletal element? Oh. It shows things coming in. That's weird. So I'll stop it and start it again. Let me just stop it and I'll restart it. If you've already done it, do it a second time. I don't know why it's not. Let me put the antenna up. Okay, here we go again. See if it does it this time. Now it'll probably double the receive. No, it looks right. Shows it receiving. That's weird.
Doesn't look like it doubled the responses. Okay, 10 seconds. 5, 4, everybody in. And wow, everybody's getting all these questions. Microtubules is correct. The other way of asking the same question is neurofibrils, and most cells are called. So anyway, they're the neural fibers that they talk about all the time that go through the axon and so forth. They're essentially microtubules. Okay, number 4. Okay, 3, 2, 1. And 93%, everybody's doing good. Yeah, it's all the cranial receptors innervate the sensory tissues on the head. So when it's cranial, it originates from skin surface or special sensory structures in the head. So it's cranial, and efferent is motor, and afferent is sensory. So very good. Number 5, last question. Which of the following neurotransmitters are exocytoles from axonal endings of the preganglionic sympathetics? Five seconds. 4, 3, 2, 1. Okay. 85. Sign of surprise. Yeah, acetylcholine is the neurotransmitter for all preganglionic autonomic neurons. So anyway, it's acetylcholine. I think we'd have all A students if I gave the exam like this and you just work together. Maybe you ought to do it that way. Yeah.
I knew you had. Okay, so now general sensory. First we have some definitions of where we're going as far as general and special senses, because she's mad at me. Somatosensory soma again refers to the body, so this is general body sensation. Tremendous number of, as the text points out, receptor systems and sensory perception or types of perception capable in this system. And that's what we're going to talk about today in the beginning of next Tuesday. Special senses is referring to the very complex apparatus, primarily in the head, for transducing visual senses and auditory and so forth. So special senses refers to very complex sensory apparatus within the skull, and we're talking about hearing and smelling and vision and vestibular senses or proprioception. Very complex area. We now know a lot about the receptors and how they work. Or somatosensory is the viscera, the sensation from gastrointestinal pain and urinary pain and things like that, as well as surface of the body. Very important clinical subject. Okay, this story begins with
transduction, we have to change the form of energy to something which we can use to sense it. A transducer is I should have my IBM fellow explain what a transducer is, but it converts energy from one form to another. So if you convert light to energy like we do with these batteries, or convert electricity to light the other way around, those are transductions, and that's what receptors do. They convert a variety of forms of energy, be it thermal or light or pressure, whatever, into a type of energy which we can understand, and we just finished talking about that. That's a nervous signal, an action potential. So these are little devices that can convert touch or pressure into action potential, essentially. A very simple example of this is mechanical. The touch perceiving ability comes from stretching neuron endings, and we've talked about before how stretch can open up channels very specifically and create changes in ion conduction, and hence possibility of signal generation. So many of these mechanical stimuli originate from an increase in sodium conduction via stretch-activated channels. And this just looks like a finger, but it's supposed to represent a nerve ending, and distortion of part of that nerve ending. It starts what we call a receptor potential, which is our next little story, which can generate or may generate an action potential, which will jump from node to node, is all that's trying to show. So what is a receptor potential? Well, that's the first part of the signal, and that's the change that occurs in the receptor in response to whatever energy form we're monitoring. And it's essentially a graded potential, or what we introduced you to before as a local potential. In other words, it's a small change in the polarity of the membrane, and it can be hyper or hypopolarization, and it's graded, which means it can be changed in range, unlike action potentials that are all or none. Primarily, this is a staged depolarization of the membrane. So it's a class of local potential. When we introduced local potential, we talked about EPPs and EPSPs and IPSPs. Those are all types of local potentials. But now a receptor potential is the fourth type of local potential which we've introduced you to. And so what we have is some change, something we're sensing, be it mechanical or chemical or light, that causes a small change in the resting membrane potential. And if that's enough to reach threshold, we'll kick off an action potential and send that to the sensory cortex, where this information is interpreted. Not all of these receptor potentials are sufficient to cause an action potential, and as we'll discuss in a few minutes, this is part of the way in which we signal the strength of the stimulus. If the stimulus isn't strong enough to reach threshold, then we don't send that information forward. So a lot of weak stimuli we never realize are occurring, of course. Another important aspect of this transducer, or the receiving of sensory information, is a subject called adaptation, which is really a valuable quality that receptors have. Some receptors are very rapidly adapting and some very slowly adapting. In the case of a rapidly adapting receptor, what we do is the signal still comes, but we only pass that information forward for a brief period of time. Let's say you sit down on the chair when you came into the lecture room. Your brain's notified of pressure as you sit down, but there's no reason for that information to keep coming back up to your brain. And so that's just one of those pieces of information we receive and then we adapt to. So if this is a stimulus, for example, and it begins with none, but then the stimulus slowly increases to a maximum stimulus, if it's just a skin displacement, usually what happens is it produces a signal which gets greater and greater up to the peak. So it's just like this displacement indicator would be like if you have a lab, the displacement transducer, which we use in lab for the frog muscle experiment. the more displacement, the more signals produced. And so it's just a linear response. The responses are proportional to the displacement. The more the stretch occurs, the more signals produced. That's a very slowly or non-adapting type of receptor. Other receptors are sensitive to the rate of change or velocity. So there's no signal until there's a change going on. It produces a signal as long as that change occurs, and then it stops. And I'm going to show you some of those in just a minute. So we say it's like the slope of in an equation, dp/dt, the rate of change is signal, but not when it reaches whatever final displacement it occurs. And finally, there are some receptors that are acceleration receptors, and I'll talk about those mostly next week, but they only produce a signal when it's increasing or decreasing in velocity. So this one produces a signal at a constant velocity, but if there's no change, then it doesn't produce a signal. This one produces a signal when there's a change in speed, or the rate of change, which is d squared p over dt squared. And actually, if this was drawn right, there should be two signals. There should be an on and off signal, because both of them have a change in rate. So that's not really a correct figure. It should have a response, both changes in speed. Right here should be an off signal. I'll show you one of those receptors also. Some examples of receptors that are fast and slow adapting, and I'll talk about this more when we get to olfaction, but one of our most rapidly adapting receptor systems is smell. Probably the people who work on our garbage trucks are happy about that. But if you're around foul odor or any type of odor for a while, you usually adapt very quickly within a few seconds and don't smell it. For example, if you're cooking a meal, you often don't even realize it. You step outside, walk around, water the plants, whatever, you come back in, that smells good, but then it's gone again. So anyway, it's a very, very rapidly adapting sensation. An example of slow adaptation, much to the chronic pain sufferers upset, is just never quits. If you have a headache, it just goes on and on and on. So slow adapting type of response is pain. Another general subject we refer to off and on is the receptor fields, how large of a field a given sensation has. And this can be interpreted a lot differently depending on what you're talking about. If you're talking about touch receptors, a given touch receptor, and mapping how far out from that receptor it perceives distortion, it might have a limited circle, and you got all these different receptors, each with their field of reception. We're going to talk about this in more detail in just a minute. So that would refer to a touch receptor's field. But if you're thinking of, and often what receptors do is very important, is precisely place the type of sensation in an area. Often interneurons inhibit the area surrounding it. So pain does this often. Collaterals from pain receptors will inhibit the area around the pain sensation so it can be placed more accurately. So often the receptive field is exaggerated by both positive or negative influences. And this is, so if you have an area that's undergoing pain, a burn or something, often the area around that will be inhibited. And if you even touch that area, the person won't be aware of it because collaterals from the pain receptors inhibit all the area around it, so it can more exaggerate where the response is. There are other classes of receptor fields, like the visual field refers to the whole visual sensation, and we'll measure that in lab that we have coming up, but we have a way to measure how far back you can perceive things in your visual field, and it's probably about 220 degrees, something like that, the whole visual field, a little bit more than 180. So it means kind of different as far as, you know, in different types of perception, and we'll talk about that as we go through different areas more specifically. Most types of sensation are controlled or it's not the right word, are assessed by converging circuitry. In other words, we have receptors coming into a general neuron that's going to go and transmit that. We call it first order and second order and third order, but the first order neuron that has a receptor often coalesces or converges on one neuron, and often there's even additional converging circuits. So a converging circuit takes inputs from several different receptors and converges them. That decreases the specificity of the receptive field, but it amplifies the signal. So let's say you had a very, very small signal. If that might not be able to be perceived the way the system is, but if we amplify that by having many of these reach threshold on the secondary neuron, you'll amplify the signal. It also means that you conserve space in the brain, and we'll talk about this later. So there's, we only have so much cortex, cerebral cortex, so part of the way of saving this information space is to converge it into and use some of the surface for very more important areas. So convergence is a very common thing in sensory physiology. Divergence is far less common, and we'll talk about a couple places where that occurs, where information from one sensory input may spread into several different circuits, and that very specifically occurs for restricted areas. One way of understanding receptive fields and this whole converging circuitry is with something called two-point discrimination. And this is another activity we'll do in lab, but we use calipers to measure two-point threshold on a person's touch, on sensing a person's touch on the surface. So let's say we touch a person, we have them look away or be blindfolded or something, and then we touch them with 2 points of this caliper, but we separate the calipers varying distances. So if you put them very close together, the person will say they sense a single touch. But as we separate them, you'll eventually receive the patient being tested will eventually say, I feel 2 different points. And so that means you've now reached the receptive field of 2 different, receptor groups or clusters. And so we do this in different places of the body and find out there's a tremendous difference in how much convergence there is. And discrimination would be inversely related to the convergence, of course. If you had lots of convergence, you'd have very close discrimination. You'd have very exacting 2-point discrimination. This is the best way to explain that whole story. So if you put those calipers within a millimeter or 2 on your fingers, you'll definitely know that there's 2, not 1. And so there's very little convergence on your fingertips and the lips and so forth. These areas can discriminate with very precise character, a 2-point from 1 point with those calipers very close together. So here's the millimeter discrimination. So even less than 1 or 2 millimeters, which is extremely small, you can tell the difference on the fingers, and the next area of preciseness is usually the lips, which I don't see. Oh, here it is. It's not quite as precise. Great convergence occurs, and there's no reason that we have that capacity, so we don't need that information. So we have great convergence from the back and the chest and so forth, and so we can put the calipers as much as 50 millimeters or 5 centimeters apart, and it's fun to do this in lab, and the person says that's 1, and why doesn't he realize there's 2? So anyway, it's amazing how far apart you can feel that you have those calipers are when you're on the back or some of these other things. So that just means that there's tremendous sensory convergence from sensory receptors in the trunk of our body. Another general concept as far as sensation is how we code the information to perceive in the brain. How do we know the difference between light, between sound, between touch? How does our brain take an information type or class and code it into anything meaningful? Well, the most important single factor is the route, the sensory pathway. As we're developing, we learn that light comes to our brains via the optic nerve and sound via the auditory nerve. So we gradually learn by matching what's going around our environment to what's going on in our brain, we learn the modality is coded by the pathway that information reaches us. There is going to be some confusion, and we'll explain those when pathways can often carry two different classes of information. But to the most part, this works fairly well, and we'll talk about some of the confusion that occurs later on when we get to different systems. So the modality is coded by what pathway the information goes to the sensory cortex by. The amplitude is another problem. The only signal we have is an action potential, and the action potentials all have the same total amplitude, so we can't increase the height of an action potential. So how do we know when we have mild pain or mild touch versus intense touch? How do we know how strong that stimulation is? Light, bright light versus very dim light. How do we code the intensity of a given modality? So if we have a maximal stimulus of, say, 100%, how do we perceive that? What type of information helps us know the amplitude of the signal? Well, there's several different ways. The first is threshold, as I said when I introduced the receptor potential idea. If the signal is so weak that it doesn't cause the primary nerve, the first degree neuron, or first order neuron to reach action potential, then we don't have a signal. So the very weakest of signals in each modality we won't perceive because it's below threshold. The second way we can do it is by increasing the frequency of the action potentials. We can, instead of sending one action potential, send many. So many different modalities are coded by how fast action potentials are received in the cortex. And another way is how many receptors are activated. Especially pain, for example. It's pretty easy to see that if we have a pinprick, we have one level of pain, but a general burn is far more. So both cases are received by the same pain receptors, but many receptors are involved in burns versus pinpricks. And so another way of realizing how intense a given modality is how many receptors are involved. And a very interesting fourth way, which we understand amplitude, is the class of receptor. And you'll definitely see this when we're talking about the cutaneous receptor family. There are some receptors that are deep in our skin, some superficial, and the deep ones are there because if we're really, get hurt severely, they're activated, otherwise they're not involved. So because of the location of some receptors, because of the sensitivity of some receptors, we can discriminate between low amplitude and high amplitude stimulation. So definitely a very important way that we code amplitude. Now I keep talking about different order of neurons, so I can finally talk about the series pathways that are involved in sensory coding. So the first order neuron in a sensory pathway is the one with the receptor at the end of the dendrite. So the receptor's out here, for example, in the skin, and the first order neuron has its cell body in the dorsal root ganglia, which we haven't defined yet and won't until the beginning of the fourth unit. But it's a chain of ganglia that goes along the spinal cord that we'll talk about very shortly. But anyway, the cell body, and it's a unipolar neuron, as you see, with the one where the cell body steps aside. So here's the dendrite portion, here's the axonal portion, and so that's the first order. The second order neuron has its cell body within the spinal cord. in a relay nucleus, and right there, decussation occurs. And most of our sensory pathways are received on one side of our system and then perceived on the opposite. We call it decussation, or the information crosses the spinal cord and reaches the brain on the other side. And we'll talk about that, and also often efferent traffic does also. So the second order is the neuron that decussates with the cell body and the spinal cord. The third order neuron, usually in sensory pathways, almost 100%, has its cell body in the thalamus. And you may not know what the thalamus is, but it's a very dark area. We call these nuclei in the core of the cerebral hemispheres, and these dark areas, or dark matter in the brain, are where you have clusters of nuclei, and like the thalamus, are often relay stations. So the cell body is in this cluster of nuclei in the center of the cerebral hemisphere. So that's the third order, and then it sends its axon out to the cerebral cortex, to what we call the sensory cortex. And in some cases, there's a fourth order. It's not often, but sometimes there's a fourth order neuron that goes along the cortex to another area. But there's almost always a chain of three, or a pathway of three neurons from the receptor to the sensory cortex. Okay, so now we can get to more specifics. That's kind of the overall introduction to sensory physiology. And now we'll get to more specifics about what we call somatovisceral sensory system. The chapter on Bohren and Volpate that contains this information has the total of sensory physiology, and they put the special senses first. So that's it. At the end of the chapter, you'll find the pages on this material, which is called general sensory or somatovisceral sensory. What we're talking about is several different modalities. The first is touch and pressure, how you know when you're touching something or someone's touching you, or pressure is pushed against your surface. Vibration is another important sensation when your car is falling apart as you're driving to work, and you feel that vibration. And finally, a very interesting sense of position. When your eyes are closed, you can pretty well sense what position your limbs are in, but it's even more important than that. For example, when you're falling, this is how we perceive that sensation. We can have reflexes to respond. When you're in the car, you can have changes in velocity or perceive and so forth. So we'll talk about this. There are a whole variety of receptors that sense proprioception, and we'll discuss them in various times in the class. And finally, we even have receptors in our joints that sense movement, and can often use those to integrate the movement with the cerebellum and make it more precise. And we have temperature receptors. We'll talk about those today if we get to it, that perceive temperature or ambient temperature, that help us respond not only by reflexes but also by behavior. We have both body core temperature receptors as well as surface temperature receptors. And finally, my favorite and everybody's favorite receptors, pain receptors. We'll talk about them and their function. The satiety of meals is one way of perceiving visceral distension, is when our stomach reaches a certain volume, but also the visceral distension is useful for bladder function as well as for controlling the movement of our chyme, we call it, digestive contents through the gastrointestinal system. Okay, so we're going to start talking about the cutaneous system and the receptors that are in the skin for that purpose. The mechanoreceptors are the receptors which we use for pressure and touch sensation. They respond to distortion or distension, however you want to think of it, stretch of the skin. And there are some that adapt fast and some that adapt slow. So I've got a table at the end of this, but we first usually go through each receptor and discuss what it does and where it's located. Then at the end, we give you a table to summarize the whole thing. Glabrous skin is skin that does not have hair follicles in it. So, I see this is backwards. Yeah, no, I did say it right on the book. So glabrous skin is like the palms of your hands and the soles of your feet. There's no hair follicles. Non-glabrous skin has hair follicles. And unique to non-glabrous skin is fibers that are actually wrapped around the hair follicle itself that are sensitive to touch. So we call them hair follicle receptors, great name. And they're the most sensitive of our touch perception mechanisms. Even just wafts of air going by our skin can be perceived by these receptors, or some people can perceive someone even moving one hair on your surface. So very small receptive field, it's just the area of that hair follicle, and extremely sensitive perception. So, and it's fast adapting. It occurs very, very quickly, and if it keeps occurring, we don't usually perceive it. Also, superficial in the dermis, another very sensitive receptor system are Meissner's corpuscles, which are simply a free nerve ending that's wrapped in a corpuscle, they say, and it's located very close to the surface. These are in glabrous skin, so that would be on the palms and the soles of the feet. Again, the more surface-oriented are the receptors, the more sensitive they are, and usually these receptors that are also close to the surface have a small receptive area, where the deeper receptors have a larger receptive field. So both of these cutaneous receptors have very small fields of reception, so you can very precisely tell where the pressure or touch occurred. Deeper in the skin, way down in the, or even below the dermis, they call it subdermal, is an interesting receptor called the Pacinian corpuscle. And it looks like this. It looks like an onion if you transect it. It has many, many, many layers of membranes. And it's quite a bit larger than most of the other receptors and deep. And these, because of the depth, and there's also fewer of them, they're not very sensitive at all. But they're mainly there to sense very heavy pressure. This is when you really get slugged by your significant other, I guess, or when you've, somehow hurt yourself very seriously. And they're also interestingly sensitive to vibration, and that's the reason is tried to be illustrated here. So many, many layers, and the nerve ending is right in the middle of all these layers. And so if you compress the surface fairly vigorously above this receptor, you first have to compress all those layers, but if it's enough pressure to compress the nerve and stretch the channels, you'll end up with a receptor potential. So you have to have a lot of distortion to occur, but and also there's some layers, some lag time. But anyway, that causes the initial signal. But if the compression remains, it's kind of a gel matrix. The gel redistributes itself, and the nerve fiber gets back to its normal orientation. So you have an on signal when the compression occurs, but if you maintain compression, there's no more signal. So it adapts to the continued pressure. But then when you take the compression off, then you'll stretch the nerve again. So it's an on-off signal as shown right here. So here's the pressure being applied. The Pacinian corpuscle produces action potential, and then when you take the pressure off, you get another signal. So it has the classic on-off type of response that you could need for things as sensing vibration and so forth. So that's Pacinian corpuscle. Large receptive field, primarily because they're very deep in the subdermal area. There's also some very slowly adapting receptors. One of them is Merkel's endings. These are only in the hands and the palms again, the glabrous skin. And they're also close to the surface. So light touch does activate these, but you'll get a continued signal, unlike the previous discussion. These continue to produce a signal for a period of time. But a small receptive field, again, because they're close to the surface. And the other type of mechanoreceptor is called Ruffini's ending, that looks like a spider web, but these are deep in the dermis, unlike the Merkel's endings, and they're sensitive to stretch and pressure more than light touch, and large receptive fields. So again, if you understand the placement of these, it makes sense. The surface ones have small fields, the deep ones have large fields. So anyway, then we have, a whole family of skin receptors that allow us to know light versus deep, vibration versus, we haven't even talked about pain, but you can see that we have to have huge numbers of these different machines in order to know what's going around us. We put all these into a figure that ties the story together, whether they're fast or slow, the fields, whether they're large or small, and the type of perception that each receptor can produce. Temperature is another class of perception that we are capable of. We have, classically, all thermoreceptors are slow adapting, which means that if we're cold, we'll stay cold, or if we're hot, we'll stay hot, even though we stay in that environment. It just takes a long time for us to adapt to it, if we even do. They spontaneously discharge, which just means they're active all the time. And we have two classes. We have a class on the surface of our body of cold and another class of warm receptors, and this is the sensitivity of the two classes of receptors. And you can literally map these. You can take a cold probe and touch it around your hand, and you'll just feel touch until you find one of these cold receptors, and it'll feel cold. So there are specific locations in your skin where you feel cold, because that's where those receptors are, and other locations, if you take a hot probe, where it'll feel warm. So there are specific receptor families and specific locales. The way which we feel just right, kind of in between hot and cold, is right when both of these families are being activated. So in the middle of this whole cascade is where our comfortable temperature usually is. You'll notice they don't say hot and cold. I ever so often slip, but they usually say warm and cold, because heat activates pain receptors, which I wanted to point out here. So when we're extremely hot, we don't really rely on those warm receptors. Pain receptors are activated, and you've done that when you burned yourself at the stove or with a match or something. It doesn't feel hot, it just hurts. And the same is true with extreme cold, and they have that on the far left here. Extreme cold is also pain and isn't perceived as cold anymore. If you've been up north and been outside shoveling and your hands get so cold that they don't feel cold anymore, they just really, really hurt. The extremes of both hot and cold are received by pain receptors being activated, not this temperature receptor system. So I think that's where I hope to get to today. So next time we'll finish the story on the sensory